this video, to get to credit, as usual, I'm going to need to write down every attendance word I say. So throughout the lecture, I'll give a set of random words or phrases. You'll need to write those down. Also, every time I say one of those terms, you'll need to take a screenshot. So at the end of it, you're going to email me a list of attendance words and a screenshot to go along with each one of those attendance words. And the last word for attendance, even if I forget to say it, which hopefully I won't, but at the very end, the last thing you should send, and you won't need a screenshot for the last one, is you need to ask me one question about the material presented in the lecture. So that's it. The first word for attendance, at least for you watching the video, is going to be dark. It's still dark outside. Anyway, on a related note, it is about 7:25, so the lecture will not start officially for another 35 minutes, so probably safe to skip ahead about 30 minutes. I will start giving course announcements and reminders before we actually officially start a date. But yeah, if you were to skip ahead 35 minutes, you would be in time for the lecture. Say if you skip ahead about 30 minutes, you'd be in time for the announcements and reminders. That's it for now. Morning.
We're in. I came in late.
So the first one here, well, first student here.
Talking.
People are dumb.
Huh?
The people are dumb. It's just dumb.
What? What happened?
There was a permanent marker up here, so I guess people just thought, hey, it's up here, so I'm gonna use it. People just don't think. No one thinks anymore. They just do stuff. Oh, my goodness. Exercise, exercise, exercise.
Jesus, where is everyone?
I don't know. Resting up for the lab, I guess. Recessive, recessive, dominant, dominant. They were all recessive. They were dominant now. So, Okay. Yeah, less, a little bit more than 3 minutes. So go ahead and start with the usual reminders and announcements that you hear every morning. So yes, attendance is mandatory. I have to turn it into the university as I did yesterday, our midterms. Participation is graded. So generally speaking, for now, if you're here, you just get your points. You can get extra points if you send in the attendance words when I'm done, as long as you send them before 9 a.m. Of course, if you're watching the video at a later date, like I mentioned earlier, you have to send in those words, you have to take screenshots. And if you're online, and I don't think anyone is right now, but if you're online, you do have to send the words. You don't need screenshots if you're streaming with us, but you have to send them in. Yeah, so like I said, participation is great. And be aware, things might change. Maybe at the end of it, I'll change it, or maybe I'll change it up for future participation points. You have to send me an email at the end of it. And I might say something like, ask me one question. Or I might say, today, you're all required to do the participation words. So be ready for me to call audibles, because I feel a lot of people aren't paying attention. Like I can see it, like when I'm grading the pre-labs, for example. Like I've been mentioning it for a week. Hey, here's what you're gonna have to do with this pre-lab. You're gonna have the video questions, and you're gonna have the introduction questions, then you're gonna have sets of hypotheses questions and tables. And I mentioned that a lot, and a lot of people missed it. And I get missing it, but not after I say it every day. So we might change things up as far as participation is concerned. We'll see. So that's all news. Any questions about that? All right. Again, to remind you, you should always be checking your emails, of course, announcements. Very valuable information there. But definitely do it before you come in, especially with it starting to, we're getting in the fall. So we might have snow days or things like that. So you want to make sure that I'm here, that I'm going to be here before you come in, because I would hate to waste your time. Any questions about that? All right. Today's lab is just as usual. So we're going to meet here at 9 o'clock a.m. Just as usual. We're going to work together as a team. So there won't be individual experiments. I'm sure as you've looked at the pre-lab, so you see what the experiment's going to be. It's going to be pretty straightforward. So yeah, make sure you're here at 9 a.m. There is no pre-lab, again, to remind you, because next week's lab is virtual. So there's no pre-lab due this Friday. So that's good news, right? One less thing to worry about. So next week, make sure you bring your computer, because again, it's a virtual lab. Any questions about all that? All right. Again, this is just a reminder. There's nothing new here. It's now too late to retake exam 1. If you saw the course announcement, you saw the statistics of how many people retook it, how many people did take it, how many people never took it in the first place. So anyway, it's too late to retake it, but you can meet me online. I will give you all the correct answers to exam 1. I will talk about why you're correct, why your answer was incorrect, and why the correct answer is the correct answer. So I give you that valuable feedback. Plus, you're gonna need the answers to the first exam because that's gonna be on the final exam. So, meet me online, get your correct answers to exam 1, and you'll also get a 5% boost to that grade. Any questions about exam 1? All right, now that exam 2 is closed, and not everybody took exam 2 either. It's now open for retake. That includes if you never took it in the first place. So obviously right now, if you never took it in the first place, your grade is a 0. So if you retake it and eventually get 100, then I'll average the 2 and eventually you'll end up with a 50. But a 50 is much better than a 0. So please retake the second exam. It's just like the first time. It's unlimited amount of retakes. It's not timed. It's open book. You have nothing to lose. Like if you get a lower grade, we just won't change your grade. Right? You have nothing to lose. It's good practice to help you for the final, right? Because again, the questions on exam 2 will also be on the final. So I don't know why people aren't retaking the exams. There's nothing to lose. It'll do nothing but help you. But again, just like with exam 1, you've got to email me and let me know. Like, hey, I'm done retaking it. I'm satisfied with my new grade. Can you please go in and average my first grade and my highest grade? So that's it. Nothing new. Any questions about that? All right, that's all I have. We'll go ahead and do attendance. Then I'll shut the door and we'll get started. Brumfeld is here. Champion is here. Gross is not here. Gunther is here. Both Hills are here. Randall is here first. Give you a little extra credit for that. Jackson, not here. Keys is here. Perez is here. Sigmund is not here. Slaughter is here. Smith is here. Stover is here in WVSU gear. Yeah, speaking of which, if I said your name and I didn't say anything about you having WVSU gear, well, I'll do it later, I guess. Tompkins is here. The Nest is not here. Watson is here. Young is here. Swyback is here. Lawrence is not here. Wilson is not here. Did I miss anybody? All right, let's get started. Turn this off. Does anybody else have any announcements or reminders? For example, I just learned on Monday that the football team was having some sort of fundraiser. That's the kind of thing when I say if you guys have announcements, you guys tell us these kind of things. We're here to help you. It's a team effort. So, we have announcements, things like that, let us know. Anyway, like I said, participation is graded. Your first word for attendance is going to be bra. Don't ask me why, but today is National or Bra Day. Bra Day USA. I don't even know what it means. I assume it has something to do with breast cancer awareness. I don't know. So that's the first word for attendance is bra. Not like my little nephew saying bro with an accent, bra, but bra. All right, let's jump back into it. So remember, this chapter is all about genetics. So we kind of started off giving the story of this guy named Gregor Mendel, who's basically, we consider him the father of genetics. He's figured out the basics of genetics. So the old textbook, which is what I'm using for this PowerPoint, the old textbook gives his story to introduce you to these concepts. So we're still learning the basics, right? You learned about the law of segregation. You learned about monohybrid crosses, which dealt with Punnett squares, which is something we're going to do in lab today. And now we're going to learn about genetic alleles and homologous chromosomes. A lot of this is going to be a review for you. Because in the last chapter, when we were talking about meiosis, remember that's when we really started learning about homologous chromosomes. Because you really need to know the difference between sister chromatids and homologous chromosomes when you're talking about meiosis, right? So you already know about homologous chromosomes. And I've already introduced this term to you, this alleles term to you. So now you're just going to learn some stuff again, a little bit of new stuff. Mostly we're just going to look at it in a different context. So here's some terms you need to know. If you did the pre-lab, you already know them. Your physical appearance is called your phenotype. At least that's the textbook definition. Of course, the definition in your lab is a little bit different, and I think it's a little bit better. Something like what is it in the lab? Phenotype is the expression of your genotype, I think is what the verbiage or the words in the lab. Anyway, The phenotype is what you see, it's what you observe based on your genes, right? So it could be your physical appearance, like it's listed here, or it could be something you cannot see, like for example, a disease, as we're going to learn about later, like sickle cell disease. You can't see sickle cell disease, but that is a result of a genotype, right? That's a disease that's a result of the genotype. And speaking of which, it's a good time to remind you, for all the labs, some people miss points on this. When you get questions on the pre-lab that are after the introduction, those questions are meant to prove that you read the introduction. So it's not just for you to, yes, I do want to make sure you understand what this word means, what that word means, and yes, you could look it up to do that, but it also serves as proof that you read the introduction. So when you're answering introduction questions, the answer should come from the introduction, just to remind you. Anyway, that's phenotype. Genotype is your genetic makeup, right? It's the genes you have. So again, using sickle cell as an example, do you have the allele? Well, and if you remember, an allele is a different version of a gene. Do you have the allele that gives you sickle cell, or do you have a normal allele, so you don't have sickle cell? Right? That's a great example of a genotype. So your genotype results in your phenotype. Genotype is responsible for your phenotype. The phenotype is a result of the genotype, right? All these different ways I can say that. So when you're studying this, again, like I usually say, don't memorize anything verbatim. I want you to understand the concepts, not just be able to spout out the words. So are there any questions between this relationship between genotype and phenotype so far? We'll go into it a little bit more. All right. So again, talking about what Mendel was doing, again, like I mentioned there earlier, each character had the same inheritance pattern, right? Because remember, he was studying a lot of different characters. And so far, the one we've been talking about the most is flower color, right? Purple or white. That's the example we keep using. But each character has the same inheritance pattern. So when he crossed the purebred parents, right? Purebred dominant, purebred recessive, whatever it may be, purple and white in the example we've been using. Once we crossed those purebreds and got the hybrids, that was the F1 generation that was the result of it. Every time, that recessive parental trait disappeared. 100% of the time, whatever was recessive disappeared. Like the example I was giving on Monday, white flowers, right? Every time, no matter how many flowers he crossed, purebred flowers he crossed, the result was always 100% of that F1 generation had that dominant trait, but not the recessive. But then, when he crossed that F1 generation with itself, he found out 100%, well, I shouldn't say 100% of the time, but a better way of saying it is, after doing that thousands of times, about the percentage he got, or the percentage was about 1/4 of that recessive trait did come back in that F2 generation. So again, that's nothing new. I'm already telling you what I said on Monday. It's just a great reminder of what we already talked about, right? So when we're talking about purebred plant mixed with purebred plant, that next generation is a hybrid. They're 100% showing the dominant phenotype. Then you cross that F1 generation, you get the F2 generation. And at that point, 3/4 of them were showing that dominant phenotype. 1/4 is showing that recessive phenotype. Any questions about that side? Okay. So the next slide I'm going to show you shows you a pair of homologous chromosomes. And again, we already know what homologous chromosomes are from the last chapter. So now we're just kind of looking at them from a different context. Because now we're not, last chapter we were just talking about how these homologous chromosomes come together in meiosis and how sometimes they have crossing over and how the homologous chromosomes line up in the middle of metaphase 1 and then how they get separated in anaphase 1, right? That's what we learned about homologous chromosomes in the last chapter. In this chapter, when we define them, we say homologous chromosomes are chromosomes with allele of the same genes, right? So there's a different definition in this chapter because we're looking at it from a different point of view. We're no longer worried about how they're traveling along in the cell when it goes through meiosis. Now we're worried about their genes because we're learning about genetics, right? So again, homologous chromosomes are chromosomes with alleles of the same genes. In other words, I keep giving random off-the-wall examples that probably aren't even true. Like, I don't know, if having hairy armpits was a gene, and I don't know, if my grandmother had the gene, the allele for hairy armpits on her, 17th chromosome towards the middle, that means all humans would have the gene for hairy armpits in the 17th chromosome toward the middle, right? That's what makes a homologous chromosome a homologous chromosome. We have the same genes in the same location. Unless something's wrong, right? For any human on Earth, that unless there's something wrong with their genome, go to their chromosomes, look at a specific place, see a specific gene, and then every other human on Earth, all 8 billion of us, would have the same gene in the same location. Now, granted, as you learned earlier when we talked about alleles, there might be different versions of that gene, but it will be the same gene, it will be in the same location. That is what a homologous chromosome is. Which brings us to a new term that I don't think we've used before. And we've basically been talking about it, but now I'm giving it a word, and the word is gene locus. That is the specific location of a gene on a chromosome. So like I was just saying with that off-that-wall example, the armpit gene, the hairy armpit gene, located towards the middle of chromosome 17, which is something I completely made up. But what I just described there was the gene locus, right? I told you what chromosome number it's on, told you where on the chromosome it is. That is a gene locus. Of course, in real science, we don't just, it's not that vague. We don't be like, yeah, it's towards the middle of that gene or that chromosome, or it's towards the tip of that chromosome. We actually have numbers. It's a little, obviously, as you might imagine, it's a lot more technical. But for us, in this 100 level non-biology major class, the way I described it is fine, right? We're just describing where the gene is on the chromosome. That is the gene locus. Again, these two kind of say the same thing. Chromosomes, they're homologous chromosomes, chromosomes with alleles of the same genes. Like I was saying earlier when I was describing that, the alleles of a gene reside at the same locus on the homologous chromosome. And that's not just for humans either. That's all sexually procreating organisms. Anything that has these numbers of chromosomes, if you find a particular gene at a particular chromosome on a specific spot of that chromosome, then it's like that for the entire population, right? Again, obviously sometimes things go wrong and it's not 100% the case, but that's usually the case. And again, finally, as I mentioned already, just pointing it out again, these alleles are these gene locuses, these specific spots on the chromosomes. They may bear identical alleles or they might have different versions, right? They might have different alleles. Any questions about this slide before I show you a visualization of all the words on this slide? All right, here's a pair of homologous chromosomes. I don't know which one it is. Let's just say it's chromosome number 5. Well, it doesn't matter, right? So here's chromosome number 5 that this person got from their mom. Here's chromosome number 5 that this person got from their dad. If we were to look at that chromosome and like, all right, let's study gene P. Let's look at their genome and study gene P, whatever gene that may be. Who knows? This is some made-up gene. If you were to look, right here on the chromosome is where gene P is. There on gene, I'm saying hypothetically chromosome number 5. Therefore, if you go to the chromosome number 5 that the person got from their dad, at the same location, you will find gene P. In this particular example, both from the mom and the dad, they both got a dominant version of the allele, right? Capital P shows that. Therefore, for gene P, this person is homozygous dominant, right? They have 2 of the same alleles, and they're both dominant. Now, if you were going to study gene A, whatever that is, some hypothetical gene, gene A is located on, again, I'm just making this up, chromosome number 5, right here towards the middle. So on the mom's chromosome, that's where you'll find the gene, just like you would for any other human. In this particular example, mom gave this person a recessive version of gene A. The dad also gave this person a recessive version of gene A at the same location on the same chromosome. And that is called a homozygous recessive, right? So they have 2 small letters because they have 2 of the same alleles and they're both recessive. Then finally, the last example, here, this person on chromosome number 5 towards the tip on their mom's, the mom gave them a dominant version of gene B. Whatever this hypothetical gene is, doesn't matter. Mom gave this person a dominant one. Dad gave this person a recessive one on the same location, on the same chromosome. Therefore, this person is called heterozygous, right? They've got big B, little b. Makes sense, right? Hopefully all that makes sense. I remember what confused me, maybe because I wasn't paying attention. I was talking about you guys not paying attention. But I must have missed the lecture or something. The first time I saw a picture like this, it confused me because I didn't get the lecture and I hadn't read the chapter like I was supposed to. And I was like, all right, this thing is pointing here saying allele, but then this thing's also pointing here saying loci. So what is it? Is it an allele or is it a loci? So just to remind you, just in case you weren't paying attention like I wasn't, and just in case that picture's confusing you. Gene loci, that's, plural for locus. So all this is saying is like, this is the location of gene B or P. This is the location of gene A. This is the location of gene B. All this is saying is that, all right, here's a dominant version of that gene. Here's a recessive version of that gene. So please don't let that confuse you. Maybe I was just a dumb student, I don't know. But yeah, for some reason that picture threw me off for a little bit. I don't remember how long. I think I figured it out before the exam, but yeah. Anyway, any questions about that? All right, let me make sure we're still recording. I haven't heard any noises from the thing recently. And come on. Yeah, we're still recording. Okay. Anyway, I'll get back to this. What are we at here? Man, I got rid of it. Sorry about that. Technical problems. Okay, so all that was just finishing up this discussion of Mendel's law of segregation. And to remind you, the big picture of the law of segregation, all that's saying is that you've got these homologous chromosomes. So that means for any given gene, oversimplifying a little bit, but for any given gene, you've got two versions, right? You've got the one from your mom, the one from your dad. What we're saying in Mendel's law of segregation is those things are going to get segregated when you produce sperm or egg, right? So when all your sperm or egg, you're only going to have one version of that gene, right? You only have one homologous chromosome for each pair. That's what the law of segregation is. Now we're going to move back to the law of independent assortment. And we already talked about this last chapter when I gave you those astronomical numbers, when I was saying in metaphase 1 of meiosis, you had all these chromosomes lined up, all these homologous chromosomes, right? Like number 1, those two versions are lined up together. Number 5 are lined up together, whatever it may be. They're all lined up together, mom version versus dad version. But they're independent of each other, right? It's not like all mom's chromosomes line up on one side and all dad's chromosomes line up on the other. Instead, it's random. So that's how we learned about it in the context of the last chapter. Let's look at it in the context of this chapter. First of all, to study this, we would do something called a dihybrid cross. And there's probably not going to be many questions, if any, about dihybrid crosses on the exam. You're not going to have to do any dihybrid crosses on the exam. You will have to do some monohybrid crosses, i.e. Punnett squares, right? You're going to have to do those basic Punnett squares that we're going to do in lab today, right? We just have 4 possibilities. So a dihybrid cross, it's not looking at just one character like we did with this monohybrid cross. It's looking at 2 characters, right? So when you look at 2 characters, it makes things a little bit more complicated. Now, it would be simple if this happened. Now, this is not what happens, but if there were 2 genes, or if the genes for 2 different characters were inherited together, then things would just be the same, right? You would still have, the F1 would still produce 2 types of gametes that they received from the parents, and the F2 would still show a 3-to-1 phenotype ratio, meaning, once you got to those great grandkids, so to speak, that last generation, the F2 generation, you would have 3/4 of them would be showing the dominant phenotype, 1/4 would be showing the recessive phenotype. That's if they were inherited together. So imagine, for example, and this is not the case, but it's easy to visualize. Imagine if the gene for skin color was right next to the gene for hair color, right? And imagine somebody's redheaded, their mom's redheaded. So their mom passes along their gene for red hairness, right? But because of that, because it's so close to the gene for skin color, they're also passing along the gene for having light skin. That would make sense, right? So that means if you got your mom's light red hair, you're probably also going to get your mom's light skin, right? Because you got her chromosome. But it's not always that way, right? Because genes are not always right next to each other on the same chromosome. And since we have 23 chromosomes, honestly, most genes are not on the same chromosome, right? They're all spread across 23 different chromosomes. So this is not what happens, right? The next word for attendance, cheese curd, because today is also National Cheese Curd Day. I don't know why, but it is. For extra credit, you could look into the biology of cheese if you want, and cheese curd. There's a lot of biology behind cheese. And again, for now, today, you guys don't have to send in the keywords, but just remember, that might change one day. I'm not gonna have a lot of warning, because I just wanna see who's paying attention. That is your hint and your warning. Anyway, that's what would happen if two genes travel together. And then again, sometimes they do. If two genes are right next to each other on a chromosome, then yes, they will probably travel together. If you get your mom's chromosome and there's two genes on that chromosome right next to each other, then you're probably going to get them, right? But again, if you got one gene on one chromosome and one gene on the other, it's a 50-50 chance. You might get your mom's chromosome for one gene, but your dad's chromosome for the other. So because of that, this is what actually happens, right? These two characters are sorted independently, meaning they're on different chromosomes. So just because you got your mom's gene for this doesn't mean you're going to get your mom's gene for that. Just because you got, I'm just throwing random numbers out here, just because you got your mom's chromosome number 1 doesn't mean you're going to get your dad's chromosome number 8 or your mom's chromosome number 8, right? You might get your dad's chromosome number 8. They're completely independent of each other. And then your book gives this example. So since we're talking about two characters, because it's a dihybrid cross, meaning two, we're talking about two characters, then there's actually 4 possible genotypes, right? There's dominant for both of those genes. Then you have a recessive for this one and a dominant for that one. Then you have dominant for this one and a recessive for that one. And then you have a recessive for both, right? Then, therefore, instead of having a 3-to-1 ratio, you would have a 9-to-3-to-3-to-1 ratio, meaning 9 of them would have both dominant phenotypes. 3 of them would have one dominant phenotype and one recessive phenotype. 3 of them would have one recessive phenotype and one dominant phenotype, and 1 of them would have both recessive phenotypes. I know that's really confusing, but this is a 100 level class, so I'm only teaching this because, well, you need to know it, sort of. I'm required to teach you. But I understand this is confusing, so as far as the exam is concerned, there's going to be very little on it. Here's a visualization of what I was saying. If these things travel together, if the gene for, all right, so R, by the way, is the gene that controls the roundness, right? Whether it's round or wrinkly. That's what the R gene covers. The Y gene is in control of the color. So if those genes were right next to each other on a chromosome, then this would happen, right? So that means if you got, right, so that means they would travel together, meaning if you got your mom's, let's just say this is the male, I'm just making that up, we don't know. If this is the male, then when it goes through meiosis, they're gonna split, but still, you're always gonna get big R, big Y, right? Because they're traveling together. And when this one goes through meiosis and those things split, you're still always gonna end up with little R and little Y, because those genes are right next to each other on the same chromosome. Therefore, this would 100% be the result, if that were the case. Therefore, when you split those up in meiosis, then you would have 50% big R, big Y, 50% little r, little y. 50% big R, big Y, 50% little r, little y. But that's not the case. Again, because they're not on the same chromosome and because they're not right next to each other, then they're completely independent of each other. So when you separate these out, there's actually more combinations. And this is what a Punnett square looks like for a dihybrid cross. In other words, as far as the exam is concerned, if you see anything on the exam and it's more than if it's more than a 3-to-1 ratio, if you see anything long like this, 9 to 3, 3 to 1, just remember, okay, that's a dihybrid cross. For the exam, that's all you need to worry about. Now, this idea of independent assortment, that is very important. But all these numbers, all this different variation, you don't need to worry about it. 100 level course. So any questions about that? All right. So the dihybrid cross that I just showed you that he did a lot of, Mendel did a lot of, it supports his hypothesis that each pair of alleles segregates independently of each other, right? So again, these alleles have nothing to do with each other. Just because you get, the female's allele for this doesn't mean you're going to get the female's allele for that.
You might get the male's allele for that because they're all on different chromosomes. And when they all line up in the middle for metaphase 1, it's it's independent. It's not all mom on one side and dad on the other. In other words, again, for independent assortment, the inheritance of one character has no effect on the inheritance of the other. So if you're thinking about siblings, right? Like you might have your mom's, and I'm really oversimplifying again. It doesn't work this simply, but it's an easy visualization. You might have your mom's eyes and your mom's hair. Meanwhile, your sibling might have your mom's eyes, but your dad's hair. And then some other sibling might have your dad's hair and your dad's eyes, right? Because those things are independent of each other. Again, I'm oversimplifying. It's not just one gene for those, but you can imagine it, right? Because of the law of independent assortment, right? These things are completely independent of each other. Just because you're getting one gene from your mom doesn't mean you're going to get some other gene from your mom. Again, unless they're really close together on a chromosome, then you're likely to get them. And of course, don't even get started on crossing over, because that throws that whole argument out the window too. But anyway, that's Mendel's law of independent assortment, right? The independent assortment is, that's the law that we just got finished talking about. Let me give you an example. Using dogs, I'm not going to ask you any questions about this example, but these are Labrador retrievers. In labs, the black color is dominant, the brown color is recessive. Being able to see is dominant, being blind is recessive. So, if you were to do the same kind of thing I just showed you with the peas earlier, where you have a purebred black dog that can see, and a purebred brown dog that cannot see, that's the parental generation. If you were to cross them, right, 100% of their offspring would be a mix of those, right? That would be the F2 or the F1 generation. And then if you were to cross the F1 generation with each other, this is what you would end up with. Generally speaking, this would be about the ratio, right? You would have, well, we haven't even got to the ratio yet, but anyway, that's the results, right? And I'm also about to introduce you to a new concept, so I also want to point this out. We know, like imagine, like, look at a dog, right? If you met this dog in your life, you don't know its genome. You can tell it's black. You can probably tell if you can see or not. So you know its phenotype, right? Like, yeah, there's a black dog that can see. Here's a black dog that's blind. Here's a brown dog that can see. Here's a brown dog that's blind, right? These are things you can see, right? You can tell the phenotype. But because that does not mean you know the genotype, right? So for example, black is dominant and being able to see is dominant. So when we write this dog's genotype, we write B because we know there's at least one dominant allele, otherwise it wouldn't have the dominant phenotype. And we write big N because we know it has at least one dominant allele for that. This blind dog, black blind dog, again, it has the dominant phenotype for the color, so we know at least has one big B for the color. Now, for being blind, if it's blind, that means it's a recessive trait, right? So it has to be homozygous recessive. So we can write that down, right? We do know that. This dog has a recessive color. Therefore, we know if its phenotype is recessive, it has to be a homozygous recessive genotype. So we do know that. However, it can see. So once again, that's the dominant phenotype. So therefore, we don't know if it's big N or big N little n. All we know is because it can see, it must have at least one dominant allele, allele. Now this dog would be easy. It's blind, it's brown, so it's completely recessive. We know if it's brown, it has to be little b, little b. We know if it's blind, it has to be little n, little n. So these are some of the kind of things you're going to have to do for the exam. Not for these dogs. These dogs are just examples. But I will explain, I'll explain some situation, and I might ask you to give me the genotype. Like, I might say something like, I might tell you everything that I just told you. Like, all right, black is dominant, brown is recessive, being able to see is dominant, blind is recessive. And then I might say, all right, there's a black dog that can see. What is its genotype? This would be one of the options. That would be the correct option. An incorrect option, for example, might be big B, big N, big N. It could be big B, big B, big B, big N, little n, but we don't, It could be big B, big B, big N, but we don't know, right? If I were to describe this dog on the test, this is the only correct answer when I ask you for its genotype. Because again, maybe it'll help you if I just write it down for you very quickly. It also could be big B, Oh man, that is hard to write. Big B, forget it. I won't do it like that. The options could be, right, big B, big N, or it could be big B, big N, little n, or it could be big B, it could be little b, big N, little n, or possibly big B, little b, big N, big N. So on a test, that might be a test question. Again, you don't need to memorize anything about these dogs. If the dogs were on the exam, I would tell you black is dominant, being able to see is dominant, brown is recessive, blind is recessive, and then I might say something like, all right, this dog is a black dog that can see. What is its genotype? Right, and those would be the multiple choice question options, and this would be the correct one. Right, with all of these, right? So I might give a test question when I say the dog is black, but it's blind. What is its genotype? And you need to figure that out. The dog is brown, but it can see. What is its genotype? You should be able to guess that in a list of different phenotypes or genotypes. The dog is brown and cannot see. What is its genotype? Right. So you might get questions like that. If you need help with it, let me know. Any questions? All right. So again, not only are we dealing with these different genotypes and phenotypes, right? We're talking about being able to decipher or guess a genotype based on a phenotype. Now we're going to go back to what we were talking about earlier, which is a ratio, right? So if you were to cross that F2 generation, these are the numbers you should see. So if you had these two, where are we at? Big B, yeah. If you had two F2 dogs, or excuse me, F1 dogs, right, that first generation, and they're heterozygous for both traits. If you were to cross them, a lot of times, like if they had hundreds of puppies, then this is about what you would expect. You would expect about this ratio, 9 to 3 to 3 to 1. 9 that are dominant for both traits, 3 that are dominant for one trait, recessive for the other, 3 that are recessive for one trait, dominant for the other, and just 1 that is recessive for all the traits. And of course, that's just a ratio. If you were to cross it, usually a puppy's not going to have that many puppies at once, or a dog's not going to have that many puppies at once, right? But if they were to have puppies over and over and over again, that's the ratio you could expect. And again, it's just a ratio. So if they had, again, thousands of puppies or hundreds of puppies, then you might look at something like 90 black, 90 of these, 30 of these, 30 of these, 10 of those, right? Again, it's a ratio, not a set number. Do I have any questions about that? All right. So again, let's go back to this idea of not knowing the genotype. First of all, yes, you will get a question where I describe something and I ask you what that thing's genotype is. And again, you'll get these options. But then there's sometimes a way where you can actually figure out the genotype and stop guessing. That is using a test cross to determine an unknown genotype. So what is it? What is a test cross? A test cross is a mating between an individual of a dominant phenotype. So again, that black dog that could see, remember? I told you this was its genotype. Big B, blank, big N, blank. We don't know its genotype. We know it's black. We know it can see. We know it has at least one dominant black allele. We know it has at least one dominant visual allele, but we don't know what the other one is. Right? We know the phenotype. We don't know the genotype. So to figure it out, we can mate it with an individual that's homozygous recessive. Because anything that's homozygous recessive, you know its genotype, right? It has to be little, right? Whatever it is. If we're talking about B, like little b, if we're talking about the color of the dog. Little n, if we're talking about if it can see or not, right? We know the genotype of anything that is showing the recessive phenotype. So for example, and you might get a test question like this. If we were to, right, again, we have this brown dog who can't see. We know its genotype. Wait, what do we call it? All right, we're only looking at one character, so forget the eye thing for now. Now it's just what color its fur is. This dog is brown. That's a recessive phenotype, therefore we know its genotype. It has to be little b. Has to be, because it's got the recessive phenotype. This dog is black, so we don't know if it's big B, right? Or if it's big B, little b. So what we do is we could do like an easier version of a Punnett square to determine what the possibilities are, right? if this thing is Big B, and it crosses with that dog, then we would expect all their offspring to be heterozygous and all be black, right? Because again, if you're heterozygous, you have the one dominant allele, therefore you show the dominant phenotype. However, if this dog was heterozygous and mated with this dog, then we would expect about half of the puppies to be black and half of the puppies to be brown. So that is how we could test and figure out the genotype of that dog. This dog right here is by crossing it with a recessive dog. So again, if those dogs had puppies and any of them came out brown, even if just one, the ratio should be 1-to-1, but it doesn't always work out that way. Even if that cross had, if you had one brown puppy, then you know for sure that this dog is heterozygous. Let me ask you this though. What if, like it shows right here, what if all the puppies were black? Do you know for sure that this dog is Big B? Or, does that say for sure that this dog is Big B if all of them are black? What do you guys think? This is where it gets a little bit trickier. I'm not going